just give you a brief introduction about ourselves. So this webinar is sponsored by Vetphage Pharmaceuticals, a subsidiary of Proteon Pharmaceutical. And we welcome, it is a pleasure to have you all here for today's webinar. My name is Pooja Goradia, and I look after the marketing of Vetphage India. And today we have with us Magda. She is the Global Marketing Head of Proteon Pharmaceuticals.
Hello, everyone.
Hello, Magda. So Proteon Pharmaceuticals, it is an innovative biotech company based in Poland in Europe. It uses the precision biology of microbiome protection to improve the animal and human health. It also eliminates the unnecessary use of antibiotics. Before we begin with the webinar, to have more, for you to know more about the company and what are we going to speak about, I will just show you a video about why we do what we do. So,
meanwhile, I think I request everyone to mute their videos, please.
yeah. Kindly mute all the, just quick rules. Mute, everyone please go on mute so the speaker does not have any issue while speaking, and also switch off your videos, because or else the voice will lag behind and you won't be able to get the flow of the presentation. Thank you.
World food production is around 400 million metric tons per year and will double by 2050. Over 2/3 of all antibiotics used are used in food animal production. The World Health Organization considers antibiotic resistance as one of the biggest threats to global health, food safety, and development today. Bacteria are becoming increasingly resistant to antibiotics. We created Proteon Pharmaceuticals, a leader in the development of biotechnology products based on bacteriophages, to help reduce the risk of antibiotic resistance. Proteon's natural antibacterial products protect the health of farm animals such as poultry, fish, and dairy cows. At the same time, our products protect the health of consumers. Our products are an effective alternative to antibiotics in aquaculture and livestock farming. They are healthier, more economical, environmental, and sustainable.
Our proposal is to bring bacteriophages to the bottle. They are the oldest and the most abundant organisms on Earth.
Bacteriophages' antibacterial potential was already known at the beginning of the 20th century, but it is only now, with advancements in biotechnology, that we can fully exploit this potential. Proteon Pharmaceuticals restore bacteriophages to their rightful place as a leading antibacterial solution for human and animal health. We have developed a special platform for the discovery, engineering, and production of bacteriophages for use against bacterial infection in aquaculture and livestock farming.
Yes, our products don't cause side effects and don't increase the problem of antibiotic resistance. They are non-toxic and completely safe for use in large populations of animals and humans.
They can be used as a prophylactic measure to improve animal health. In addition, they are economically efficient in industrial application and easy to use. We think about the future of all of us.
So with this brief introduction, let us quickly begin for today's session. I will introduce with the speakers for today. We have with us Dr. Marek Rymanowski. He is the head of veterinary services at Proteon Pharmaceuticals, based in Poland, and is responsible for the corporate global techno-commercial support. He is a leading expert in poultry health and nutrition. Hello, Dr. Marek. We also have with us Dr. Ramdas.
Hello.
Is the director at Vetphage Pharmaceuticals and is responsible for sales in entire APAC region. It's an expert in feed formulation, technical services, and overall techno-commercial activities. Hello, Dr. Ramdas, once again.
Hi, Pooja. Thank you so much.
So before I hand over the mic to the speakers, let me just quickly run through the agenda for today. So we will go through the Poultry India snapshot, followed by the use of antibiotics and bacteriophages in poultry. And then we will have the future of bacteriophages in poultry. Don't worry, we are not very technical and we won't just end up with this one-way dialogue sessions. We have with us a Q&A session. I'm sure the speakers are very good, but I'm equally sure as I see the audience is also great over here. So speakers, please be prepared for good questions coming at your end after the end of the session. And we have a fun session towards the end. So everyone, please uh, stay on till the end and don't miss on the fun time. So I hand over to you now, Dr. Ramdas. You have the floor.
Yeah, thank you, Pooja, for the brief introduction of starting with the video of the company. It's my pleasure to represent a company, your products, and your technology, the Vetphage with the group. Can you take us through the Poultry India snapshot? There are many experts present in today's webinar. So thank you so much, guys. You spending valuable time. जैसा कि आप जानते हैं, I will try to mix up Hindi and English. So we are Poultry India. Who doesn't know about Poultry India? So this is third largest pig producer. We are just behind China and US. I know sometimes we are afraid to take the name of China, but what can we do? We are in the business. So we are fourth world largest chicken producer. Again, the China name is coming after Brazil, China, Brazil, and USA. We produce around 75 billion eggs per year, and we produce 4.2 million tons of broiler production. Precisely, if you look at this, so poultry industry is like a corporate sector, and it's adding a lot of value to the Indian GDP. And now this, because of this coronavirus outbreak, so people they were concerned, and now things are getting improved. So I think we should maintain and retain these positions even coming time as well. So I will take you through the small brief about the Indian market overview. About the poultry industry. See, this is we generally call this Dr. Rao, father of Indian poultry industry. So precisely, this industry is almost 6 decades old, or you can say around 60 to 70 years. And we have approximate 5 lakh broiler farmers. And these are the presented facts in various webinars, seminars. So you can challenge also, but more or less these are the same. So we have around 25,000 layer farmers. Okay, this is very important. It's 1,500 consultants or the key opinion leaders, and most of the key opinion leaders, especially the doctors, are attending the seminar. We have approximate 700 hatcheries. There are many people involved in many feed companies, the traders, the retailers, and farm staff and veterinary pharma, which is concluded something around 10 lakh people. So this poultry industry is giving the jobs, or you can say the engaging directly or indirectly to around 1 million people. So this is small snapshot about the poultry industry, and many experts are there. I should not tell this fact. So, but I need to take you through this ever-growing poultry industry, which is passing through the challenging time, but definitely it will improve. So over to the next slide, Pooja. So then I will hand over the mic to Dr. Marek. He's expert in antibiotics and the bacteriophages, and he's from the Global Veterinary Services. So he can take you through the presentation. Thank you so much. Over to you, Marek.
Hello, thank you, Ramdas. Hello, everyone. It's nice to meet with some people I see by names, like, for example, Dr. Sika. Hello, the professor. How are you? I'm very pleased to speak here to you. Here is very light subject, but very important subject, especially in poultry, but not only. So for our humanity, it's very important. So antibiotics and bacteriophages, because we need to know that we have to have some alternatives for antibiotics. And why, I will tell you shortly. So first of all, antibiotics. Discovered in the beginning of the 19th century, were taken out the first place in the race to have a possibility to treat diseases which are till that time was not cured. So discovery of the penicillin was a crucial moment to for the humanity to start fighting with the diseases which were declining the numbers of people around the world. Penicillin started this, and a lot of many different substances were discovered since that time. Why antimicrobials? Why antibiotics are so powerful? Because they have more than only one target of the bacteria. So they have very broad spectrum. They might have some broad spectrum. We are saying broad and narrow spectrum, but still comparing to the bacteriophages, antibiotics are killing many different bacteria. The way how they are doing this is also very differentiated and fragmented. Some of them are inhibiting the cell wall creation. Some of them, they are messing with the enzymatic processes inside the bacteria, but all of them are used to kill the bacteria. All of them also have some good possibilities to kill the bacteria which were untreated during the centuries on the humans and animals also. But during the years of usage, and it was let's say predicted by different scientists that a lot of germs can take the resistance against the antibiotics, and this is what is happening. And that's why all the world, and including also our company, is focusing on the alternatives from the antibiotics. Also, you need to remember that antibiotics kill the bacteria without checking if it's good or bad bacteria. So they are also influencing our gut microflora, which, as you know, and the recent researchers in the recent years are confirming that the gut microflora is getting crucial aspect of immunity, both in humans and animals. Of course, we need to remember that there is a possibility that bacteria will gain the resistance against the antibiotics, and both by using or overusing the antibiotics in veterinary and in human medicine, but also they can take the resistance against antibiotics by simply exchanging the genetic information inside their colonies. So using vectors, so using the exchange actions. And according to the FDA, which this is very important information for you, according to the US FDA association, 80% of all antibiotics sold, which is more or less around 20 million pounds, was used in the production animal. That's why we are here. That's why we invite you, veterinarians and people which are linked with the production of the poultry, because we are on the target from different associations. This is about antibiotics. Next slide. Bacteriophages. Bacteriophages were also discovered in the beginning of the 19th centuries, but ease of use of antibiotics and efficacy of the antibiotics and understanding, much more easier understand the way how they are working against the bacteria, caused that the antibiotic won this war. But now, during the time where the antibiotic resistance is a very huge issue, and when the humans cannot have a good treatment because of overdosing, overusing different antibiotics, bacteriophages are coming back. And here we can propose you our offer from Proteon Pharmaceuticals. What are bacteriophages? Bacteriophages are the naturally occurring viruses against bacteria. Yes, bacteria can have viruses too. They are the natural organism made up during the years, centuries of genetic changes. And I like to name the bacteriophages is the nature way to control bacterial population. So the nature is so smart itself that she developed the tool which is using to suppress the population of different bacteria. How the bacteriophages are doing this? They are doing the scientific researches during years. We discovered that the bacteriophages had two different life cycles, depend of the type of the bacteriophage. First cycle is a lytic cycle, which is most predominant for us. Is this the situation where the bacteriophage is infecting bacterial cell, injecting the genetic material inside of bacterial genetic factory, and changing the biohistory and the production of the genetic material to produce the copies of the bacteriophages. But they are doing this in the so sophisticated way that the result of this change is the lysis of the bacteria. So in other words, bacteriophage is killing the bacteria. In opposite to this cycle, we have lysogenic cycle. In this case, we don't face the death of the bacteria, only the bacteriophages infecting the bacteria, injecting the new genetic information is changing dynamically quicker or slower the genetic potential of the bacteria inside. This is not usable for us as a, let's say, veterinarian who wants to treat bacteria by killing the bacteria. That's why our technology in protein pharmaceuticals leads to have the possibility, or we have power to check either we are dealing with bacteriophage which is lytic one or lysogenic one, and we are choosing only lytic one. Next slide, please. In this short movie, you will see how it goes with the lytic cycle on the bacteria infection. So please put on the video. Okay, so you can see here that the bacteriophage which is attaching to the bacterial cell and start to inject the genetic materials which in general change the genetic processes inside the bacteria to start creating the copies of the bacteriophage, which releases of the lysis of the bacteria. So this is the most important information about bacteriophages and how it is in confronting to the antibiotics. We can, let's say, Pooja, please. We can, let's say, differentiate with target, accumulation, side effects, resistance, and efficacy in front of the biofilm. So with the antibiotics, the broad spectrum is the, let's say, huge plus and huge minus, depends on the situation. They don't accumulate too much in the live organism, but they are, after the use of different antibiotics, especially in human people like tetracycline, for example, side effects are very, very visible. And of course, what I mentioned, resistance can show up and is showing up very, very often. And last year shows up that there is plenty of different bacteria families like Salmonella or Staphylococcus, Streptococcus, which are getting more and more resistant to the most commonly used antibiotics. And what about biofilm? Biofilm is very hard to reach. Biofilm is, let's say, a way of protecting the bacteria against any other influence from external, from disinfectants, and also including antibiotics. So antibiotics cannot reach the bacteria which are attached to the biofilm. Comparing to the antibiotics, bacteriophages are very, very specific. It's like, you know, American rocket called Patriot, which targets only specific targets, specific cars, specific home, or specific bacteria in case of bacteriophages. They can accumulate in the environment. They are living everywhere, so it's not a problem for them. Side effects is not visible at all. We tested our bacteriophages with even 10,000 times overdosing the standard dose, and there was no side effect both on in clinical trials and also in vivo. We don't know any information about side effects or resistance build-up. If there is it goes, I think, in a more natural use. So when the bacteria is getting resistance against some bacteriophages, the bacteriophages also eliminate this, but evolve to this to start to be effective again. But in our case, we have a cocktail of different bacteriophages, so there is no chance to build up resistance against this. And also, bacteriophages, as a part of the biofilm, also can reach the bad bacteria which are attached to that biofilm. So, what is reality? So, having this in your mind, the whole world is thinking about how to solve the issue with the antibiotic resistance, and the main reason is that, as I said, 80% of antibiotics are consumed by animals, 20 are only consumed by humans. But having a possibility for having antibiotics and possibility to treat humans, we need to focus on the decrease of usage of antibiotics on animal production. Next slide. What is happening in the industry? The whole world is, let's say, thinking about the best solution to avoid usage of antibiotic, unnecessary usage of antibiotics, or overdosing the antibiotics. And not only we here in Poland and European Union. I know there are some people from European Union beside of Polish team, and that they can confirm this, but also World Health Organization or European or American Center for Disease and Control are shouting about antibiotic awareness and antibiotic resistance, and they are trying to educate people, educate doctors, educate veterinarians, educate farmers about why we should use, if it's needed, antibiotic smartly and wisely, not using this without any possible reason. So it's not our idea, it's an idea which is worldwide. And here you can see, let's say, example of one of first superbugs, which were, superbugs means, so it is resistant against all known antibiotic used to cure. NDM1, so New Delhi, which is particularly Salmonella, started in India and spread it across the whole world in few years. Look, in almost 10 years, you have this diagnose this bug across all globe. So the problem is growing. It's not only with Salmonella, it is also with the Staphylococcus, Streptococcus, and plenty of other bacteria which are getting the super resistance against antibiotics used in this in the modern medicine. And I think, Pooja, next one, please. Yeah, that's all from my side. I'm coming back to Dr. Kamabale. He will tell you about some future bacteriophages.
Thank you, Marek. This is very in-length information. And now what next? How is the future lies about the bacteriophages? How we are going to overcome these challenges like superbugs, and how this theory evaluated as an alternate solution to antibiotics, not only to antibiotics, many indirect solutions. So Pooja, if you can play on the screen, that will be a great idea. Like, guys, if I ask you the question, हम गंगा जल में क्यों नहाते हैं? Probably ever thought about. तो गंगा जल में इसलिए नहाते हैं कि गंगा जल के पानी में एक अमृत जैसा चीज होता है, वो है nothing but bacteriophages. So people go through Ganga river to immerse their body to get it cleaned by bacteriophages. I will take you through how this medicine, or you can say the use of antibiotics and all those things has been taken place. In ancient days, like 1800 and below, see, Ayurveda is the oldest science of medicine. We are Indians, and all people, they used to treat with the herbals and all those things. And there was a preliminary research just now I mentioned that people, they used to go to Ganga river and clean their body. So there was a discussion, and there was some research started happening in 1915 around bacteriophages. They were discussed, but they never come into the existence, and they were came into commercially viable. And then with emergence of antibiotics, everything got stunned. So antibiotics means Ramban Ilas. So we are the doctors. Many people here are doctors. And then in the ancient area, in the villages, so people used to take antibiotic for everything, even for fever, even for small cough, and all those things. So overuse started happening. Just now, Dr. Marek mentioned 80% overuse started happening in the animals, and only 20%. So here is the mix and match, and that antibiotics become a very predominant factor. And in 2015, or you can say early 2000 or mid 2000, so this the research started happening on prebiotics, probiotic acidifiers, or you can say the essential oils and all those things. But let me tell you guys, prebiotic, probiotics, and acidifier, you know what is the mode of action, and they are not a direct replacement of antibiotics. In any case, I can bet you, prebiotic, probiotic, they work on the competitive exclusion, acidifier by maintaining acidic pH, and essential oil by immunoboosting and all those things. So, but these are all indirect treatment. And many people, there are some products in the market, and they confuse with by using the probiotic, like this, chili, the Bacillus subtilis, and they confuse the people that this was like a bacteriophages. No, this is not. Now, after 2020, so the real research started happening, as Dr. Marek mentioned, Poland and the Proteon, they are the pioneer to get various patents for these bacteriophages. And probably I would say the Proteon is the only company is having the very, very precision technology on the bacteriophages. So 2020, I would say this will be the era, or this will be the century of the bacteriophages. Because when the overusage of antibiotic and the regular what is called FDA approved usage of the antibiotic start happening, so you need to find a solution that is nothing but bacteriophages. So I dedicate this 2020 onwards the bacteriophages, and you will witness in coming future. Next Pooja. Okay, I would like to take you now. This is a very dirty time, or very dangerous time of Corona. Now, what is happening after this Corona outbreak? So there is a paradigm shift in the demand in poultry, because everywhere, whether it is US, Europe, India, everywhere, people, they are promoting poultry as an alternate, or you can say the fight against the immunity suppression due to coronavirus. And the price started falling, now it started jumping. This industry is coming to the stability. So just look at this. Food safety is a prime importance after this coronavirus. Do you think now the butcher shops, I stay in Mumbai, so there are many butcher shops around. But if you compare with the butcher shop, chicken from the butcher shop, and chicken, you are getting it from the hypercity, or you can say Heiko, or the big malls. This is very nicely packed. Air conditioned, like Chora bean chicken or something. I don't want to name anybody's, like Godet chicken or something. So you need to get the chicken, or you will believe to buy the chicken. It is nice, hygienically packed. It is branded like antibiotic free or salmonella free. The word will come that people, they have to brand salmonella free or E. coli free. And then because of this, due to coronavirus outbreak, consumer awareness is at its peak. And some very learned people are there in this call, so they will witness that this paradigm shift is happening. I just wanted to take you through this Times of India cutting. This is a paper cutting, and it talks about superbugs. If you know, Dr. Marek mentioned that superbug story started in 2005, and from Delhi it started, and it spread to the world. It's like a wildfire. and then it becomes start with the antibiotic resistance. So you just go through this presentation, or you can see this you can find it also in Google. So there are many people died because of the superbugs. So India is under superbug threat, and it is very, very dangerous. This is the paper cutting. You will find it in Google, and there are many people died because of the food poisoning, because of the even the fever and all those sort of illnesses. So you need to take care. And after this coronavirus, definitely industry is going to witness a very, very hygiene practices, and I think we should be prepared for that. Next, Pooja. Okay, now what we did various bacteriophages trials in India. See, there are various trials. The trials happened in Europe, trial happens in US, trial happens in Brazil, trial happens in India. So I will take you to the trials we did in India, and we did on all parameters. These are basically the broiler trials. Look at this: mortality, loose dropping, FCR, immunity, food safety, and bird appearance. This is very interesting. Bird appearance. So wherever we tried through various big consultants or big doctors, and even the poultry farms. I don't want to name them, but they're the very good practitioners. They have tried it, and everywhere they got a body weight jump. I will take you the next slide how the body weight jump was. You will imagine they got the body weight increase by something around 150 grams. Just hold on. And we tried on hatchability and egg production in breeders. There is tremendous improvement in hatchability and egg production. And in the commercial layers, egg production, eggshell quality, and egg quality. Eggshell quality and egg quality. So there are two various parameters. In every parameters, our products have come to the occasion, or they stand to the occasion. Now people, they may confuse that whether there are salmonella in commercial layer or not. Believe me, the salmonella is very prominent in commercial layers, especially when it goes to the for egg powder. If somebody supplying for the egg powder, so that needs to be certified as salmonella free egg. And in that case, when the branded egg also is coming into the picture, then they have to certify that this egg or table egg is free from salmonella. So body weight improvement in the broilers, hatchability and egg production, even the mortality. We have tested in the mortality also. Some of the big integrators, mortality also came down in breeders. And commercial layer, egg production, eggshell quality, and overall egg quality. So these are the various parameters. Next, Pooja. Okay, so what was the efficacy? So we have the product Bufazal, which is the combination of various bacteriophages. And just I will take you through what was the mortality control. Just look at this. Mortality controls. It was very, very nicely managed farm, having 6% mortality, and he was a happy-go-lucky person, and he was happy with the 6% mortality. When he tried our product, so he got up to 4%. So there is 2% improvement in the mortality. So the 2% is quite high, and everybody will witness that. So next, Pooja. Body weight. This is very interesting. I mentioned earlier in the few minutes ago that the person, the big integrator, was getting a 2 kilo body weight, 2150, and he got return on investment like 1:3 or 1:4. So he spent 1 rupee, so he got 4 rupee return out of that. So can you imagine? It from 2 kilo, so 2150, so 150 gram extra body weight. So there is 8% improvement in the body weight. And this, I assume everybody witnessed that 150 gram is a quite a huge increase in the body weight. And ultimately that will, and the mortality control, the body weight increase will lead to the FCR. Now the FCR. Next. Just look at the FCR. Okay. Practical condition where everything was under control. We was very happy in practically. These were the products tried in the integration farms. And 1.6 was his regular in the FCR by using all antibiotics, his own program, various, even sometimes EC shed also. So in practically, we got 1.55 FCR ultimately because of the reduction in mortality and improvement in the body weight. So there was improvement of 3.12% in FCR. So in broilers, this is making as wonders. Broilers, nobody talks about salmonella, but now I came to know that some of the integrators, some of the farmers, they are supplying to the food chains like KFC, or you can say the big malls. and they need to certify that the chicken is salmonella free. Ultimately, with salmonella or E. coli, we are also coming with E. coli. So if we are talking about this, definitely the food safety concern, and if you have to certify with salmonella free, the broiler becomes very, very important. So I would suggest, if you are getting the wonderful performance in terms of broiler, in terms of body weight, mortality, and FCR, and also the production improvement and hatchability in feeders, and equality and mortality control in layers, this could be a wonderful solution in coming days. So this is the future lies with this product. So thank you so much, and now it's open for question and answer. Pooja, over to you now.
Thank you, Dr. Ramdas. Thank you, Dr. Marek, for the wonderful insights on the usage and overusage of antibiotics, and also for the future of bacteriophages in this poultry. It does sound very promising with the facts and figures that you stated. However, I'm sure that our expert audience will have many questions for you. So I'll just quickly go through the rules for the question, asking the questions. Everyone, please, we are open for the question and answer sessions. We already have few questions on the chat, and I will request all of you to put all the questions that you have on the chat itself to avoid any disturbance. We will read it out for you, and the speakers will try and answer to the best of their ability. Let me just warn you, speakers, we already have our chat box flooded with questions, so please buckle up. Coming to our first question. The question is by Dr. Ajay, and the question says, how many days did phage stays in gut in the active phase once administered?
So I will answer to this question. So based on our research for the thermodynamics of the bacteriophage inside the organism, we find out that usually when there is a host for the bacteriophages, they are multiplicating and staying in the organism for a longer time. If there is no targeted bacteria, if there is no host for the bacteriophages, the dose which is administered to the poultry will eradicate from the organism during next 48 hours. So 48 hours is the maximum time where the bacteriophages after one dose stay in the organism.
Thank you, Dr. Marek. I hope, Dr. Ajay, you got the answer. I mean, the desired answer to you as per your satisfaction. Now, the next question is from Mr. Shivanan. The question says, which targeted bacteria it covers, and any specific study do you have?
So Bafazal plus is working against Salmonella enteritidis and Typhimurium, and additionally it works against Caenarum. It consists with the 6 different bacteriophages, 2 for each environment. It works also against many different, let's say, call it exotic one, but each time when we are dealing with the exotic type of Salmonella, like Java or other, we need to find out this, is it susceptible to bacteriophages or our cocktail in our lab.
Thank you once again, Dr. Marek. The next question is, what are the results expected in mixed bacterial infection like Mycoplasma and E. coli infection?
So, as I said, bacteriophages are very precise comparing to the antibiotics. The spectrum of the working is limited to 1 bacteria. So, in case of having the systemic infection or mixed infection where the CFD occurs, like Mycoplasma, E. coli, and many different bacteria, it will kill only or Salmonella itself, it will kill only Salmonella which are susceptible to bacteriophages. So in this case, we suggest also to use antibiotic in this case, or wait a little bit. Maybe soon we will have some solutions also for E. coli in India. So I hope it will be a success story, same as with Bafazal. So in this case, you need to think smartly, and when you are dealing with the mixture infections, you need to also think about additional antibiotic treatment to treat the other bacteria which are not killed by bacteriophages.
Thank you, Dr. Marek. We have a lot of ongoing questions, so I'm just shooting one after the other. The next is, can we use acidifier with bacteriophages?
During our research and stability testing, we found out that some of the organic and non-organic acids, but very few, are not harmful to our bacteriophages, but most of the organic and non-organic acids are harmful by decreasing the pH. So our recommendation is to, during the administration of bacteriophages, you need to avoid in the same water usage of acidifiers.
Guys, if anyone is not satisfied with the answer, please put again in the chat box. We will try to answer it more thoroughly. The next question goes as follows. So how many bacteria species does Bufazil plus G work?
It's the repetitive question. So as I said previously, Bufazil plus G is working against Salmonella enteritidis, Typhimurium, and Salmonella gallinarum pullorum.
Okay. The next question says, how about dosage of bacteriophages? What about dosage of bacteriophages in multiple phases of broiler growth, considering perfect balancing of that microflora between 12 to 32 days of life?
Using bacteriophages as it is in the same scheme as it was with the antibiotics is useless, because antibiotics is the chemistry, the substance, and bacteriophages are naturally living organisms, which are administered by externally by us. So in this case, we suggest to avoid putting bacteriophages in case of disease, only to prevent the disease. So we suggest to administer, since day old chicks throughout the whole life, every other day, to have the full protection against different infections. So putting it is possible to administer also in case of disease, but usually the disease is the mixture of the different bacteria. So in case of Salmonella and, for example, E. coli, we'll solve issue with Salmonella, but we will not solve issue with E. coli with our bacteriophages yet. So you need to still think about common treatment together with antibiotic or maybe other substances.
Thank you. The next question says, how about bacteriophages getting resistance?
Pooja, I would suggest please name the question, who asked the question, because there are many learned people in this group, so that they should also, that will give the excitement to the other people. Like Dr. Ranade I have seen, Dr. Prajapati sir I have seen. So there are many people I have seen in the group. So
there are many questions coming in.
The questions are coming out, but I don't know if we are able to answer to all of them, but we will try. So answering to the question regarding the resistance, so they are evolving. So in case of bacteria which are getting in this or other way resistant against antibiotics, the same way the resistance is getting against bacteriophages. And also bacteriophages are live organisms, so they are also evolving, they are also mutating, they are also changing, especially when there is a lysogenic trials or also lytic trials, which can lead to changes of genetic material in bacteriophages. But in case of resistance, we have some, I believe, good approach from our side. We put it in our product not only one bacteriophage against Salmonella enteritidis, but two, against Typhimurium 2 and against Gallinarum 2. So they have no chance to develop the resistance against both bacteriophages, because those two different bacteriophages have different points of attachment and different genetic materials, which it's impossible for the bacteria to in short period to develop resistance. So yes, it is possible in the nature, but it needs to have a very long time to do it.
Thank you, Dr. Marek. So as Dr. Ramdas said, the next question is by Dr. Prajapati. He says, are bacteriophages effective for systematic bacterial infection? If yes, then what about immunity against the bacteriophages?
Bacteriophages are, very common in the environment, inside and outside organisms. So they are they are viruses. So they are external compositions. So they are treated by let's say external antigens. In our researches, we found out that there is, and not only ours, but also other scientific university scientific documents is saying that bacteriophages can interact with the interleukin system of starting or depressing the immune response to the particular antigens. So from one side, they can block or they can hide from the immune system. And from other side, they can stimulate a non-specific immune system to be as a first, let's say, tool to fight against the infection. So in case of systemic infections, there are some scientific reports showing that bacteriophages, if I understand you right, Dr. Prajapati, they can cross in some percentage of the dose, they can cross barrier blood-intestine and go into blood system to the every internal organ they can reach with the blood. So yeah, I believe that it needs to have a bit longer or maybe a different dose of the treatment in this case. But yes, it is possible to treat bacterial infection. We found out some in vitro trials also that our bacteriophages administered orally were found out also in blood and internal organs. So yes, it is possible.
So the next question, just a quick update, guys. We don't want to take more of your time than what we asked for. So we just have, we'll take for more questions for just 5 minutes, and then we won't miss out on the fun session towards the end. So don't worry,
I believe that we will collect the remaining the questions and and we will answer, let's say, offline for this, and we'll send to all participants with the prepared answers for this.
Yes, we'll surely answer each and every questions and send it to everyone.
Okay, so 4 more questions, okay, and we'll shortly finish.
Okay.
Yes? So how to preserve bacteriophages? The next question from 6Pro, how to preserve bacteriophages. I don't know what is behind this question, but I can tell you that first researchers and first products containing bacteriophages were suffering because of the especially preservation, also stability of the product. Our products gained very recently 2-year expiry date, which we guarantee that they are still positive, they are still working. So it's part of our patented technology. It is possible to preserve bacteriophages. It is possible to have them at high quality for more than 2 years. So yes, it is possible. I know that some products which were across India were suffering because very short period of shelf life. It was around 3 or 6 months, if I believe. So yeah, I understand this question if it lies behind. Yeah. And yeah, next question from Dr. Renade. Do you think that the specificity of bacteria can be a problem in the field condition? Yes, but I believe that we are just on the beginning on the road. I mean that we focused on the developing the product against Salmonella. We shortly, and Dr. Ramdas, please add a few words from your side. We are shortly preparing to launch a product against E. coli. Dr. Ramdas?
Yeah, so sir, Dr. Anandi, sir, thank you for your question. So this trial has been taken place in Kolkata, and we are trying. Actually, this product is already tried in European condition, US condition, but we wanted to test it in the Indian condition because Indian condition is absolutely different from other conditions. And then whatever we are trying that should work in Indian condition, probably by a month or so we'll be ready with E. coli solution, and that could be. And as per Dr. Marek's and the Protean's assumption that India should be free from E. coli. If that happens, so that could be a wonderful achievement, I would say. Because E. coli is bigger problem as per perceptions of the doctors, as per the actual happening in the field. So people, they are worried about E. coli. Hopefully, this should be a permanent solution in coming future.
And a question from Professor Sika. Hello, my friend. My suggestion, your product Bafazal can be positioned as oral vaccine, and what is our opinion? Yes, it can be considered as a kind of vaccine because we have some theory which we are now checking. Imagine the situation when bacteriophage is killing the Salmonella bacteria. So when it's killed, the bacteria remains act like an antigen in organism. So immune system of the organism is starting to produce immunoglobulins against those antigens. So this is the same way as a vaccine is working against. So yes, it can be considered, but we are facing. Yeah, it's time to pray. I know. It's more problematic with the registration of the product with such a use. The easiest way it was to do it with the feed additive. Any other, let's say, category of the products pushes on us a lot of more effort to deliver to the registration authorities to have the possibility to have this product as a disinfectant or the vaccine. So I don't know, it's still open, I think, for the
Thank you, Dr. Marek.
Marek, we will answer this question to Dr. Sika separately. Thank you, sir.
Thank you.
Thank you.
I think there is a question from Dr. Abhijit Mishra. So can you read that question?
Which
Dr. Abhijit Mishra from Kolkata.
I can't see this.
It's the last one.
The last one. If you use bacteriophages without water sanitation, then how we can prevent waterborne during the time of the bacteriophage treatment? Bacteriophages are very tough products, but they cannot survive the working solution or the base solution in the water tanks. So we advise not to administer it in the same water when you administer the disinfectant or acidifiers. But for example, we tested bacteriophages in front of the very high concentration of chlorine, and guess what? They survived and they were very active still. So we can say that there is a possibility to use still disinfection, but our suggestion is not to put this into the base solution during the preparation of the administration. So it is possible. With the waterborne, I believe that we should approach in complex ways. So first of all, identify the problem and identify the source of the problem, because if it's still from the water, why it's in the water? Because of cross-contamination, maybe some other stuff. You need to look at this in this way, I think. And when bacteriophages, sooner or later, as I said, we are expanding the portfolio of the bacteria that we can fight with. So let's give us some time, and we will come back to you, and we will have maybe solution for everything. Who knows?
I think Pooja, that's the final question, and then we can answer them separately with email or something.
Yes.
So then let's move to the fun activity.
Yeah, when I was thinking the same, maybe we have had too much of technical. Now let's move to the fun session. So now, I'll just quickly tell you about the fun session that we have. So we'll be asking 5 questions which have multiple choice answers. So you just have to choose the correct one. I'm sorry. You put the answers in the chat box in the format that has been displayed on the screen. So just an example, maybe the first question answer is say.
You need help from Magda? No.
I'm sorry. I was just, I guess my throat was not good. So I hope I was clear. You
Yeah.
just have like 5 questions. We have multiple choices. You just put the answers in the chat box, and here we go.
So please kindly give your answer to the chat box, stating number of question and the answer that you think is correct.
So what is the prize for that, Pooja? Because the quiz winner should get the prize.
We have interesting Amazon vouchers coming their way. Whoever answers the first 5, the quickest first 5.
Please give all the answers in one message.
Like this, B, C, D, A, B, like this, right?
Yes, all the answers in one message, please. And in a few minutes, you will get the correct answers. Please
Only put the words in sequence, like.
Please, yes, please kindly give all the answers in one chat message. The sequence of letters.
Yeah. No need to put 1, 2, 3, 4. Just A, B, C, D, A, something like that. You can put it like, yes, this is.
Exactly, that is how we do it. So we will wait 2 more minutes for everybody to have a chance to give their answers. And we will
I'm excited to know the top 5 to get the best answers.
One more minute, and we will close the questionnaire, and we will show you the right answers, and let everybody know who the winner is. Okay, I think
Time up, right?
I think the time is up. We can check the answers.
So A, C, B, C, D, right?
A, B, C, sorry,
ACBCB.
ACBCB. So there is
So how many are correct?
correct answer from Dr. Mahendra.
Wow, clap for him.
Dr. Kishore.
Great.
I can see Ujjwal Dalal.
Thank you, Ujjwalji. Congratulations. You are
Jaguru Kadam.
Jai Guru Kadam. Great. Kadam Sahib, namaskar. I think you name everyone who are right. If you can.
Okay. So
I have a private message from Rajaram and he's correct. ACBCB.
Okay.
okay. So we will contact you in order to deliver you your prize. Congratulations. And once again, thank you for joining our webinar. This is the first one. We will be inviting you shortly for the next one. And
Dr. Obi Mishra is also correct.
Who?
Dr. Obi Mishra.
Abhijit Mishra, sir. Great. You put down Pooja, Dr. Abhijit Mishra, sir.
we have saved the chat, and we will double check after the webinar. So we will not miss anyone.
Great.
Consolation prize to everybody.
Wow, Dr. Sikka, consolation prize.
To everybody, just to motivate for next time.
Okay.
Thank you for suggestion.
Yeah.
Okay.
So everyone, thank
Professor
you.
Sikka, we will think about this. Thank you.
Okay, thank you.
Thank you, Dr. Sikka.
So,
Well done, Malgada, well done.
let me conclude. So thank you participants for your active involvement in this session. Thank you the organizers. Thank you Dr. Marek and my team and the veterans who attended this seminar, like Dr. Prajapati, Dr. Anade, Dr. Sikka, and everybody. So that you are our strength and you stand behind us. This is a novel technology and this is going to change the world. As I said you, as 2020 is going to be a bacteriophage world. And as our E. coli product is coming and already we are established in the company, so this will be an ultimate solution as an alternate to the antibiotics. And coming future, as a food safety is increasing after coronavirus, this is going to be a win-win situation for everyone. Thank you so much, guys.
Thank you again for participation. We will share the recording shortly, and we will also get all the questions that you have asked on chat, and we will send you all the answers. So everybody is satisfied.
Great program.
Thank you so much.
Congrats to Proteon and Vetphage. Thank you.
Thank you, sir.
Thank you, everyone.
Bye. Dr. Marek.
And have a good afternoon.
Thank you.
Thank you. Bye-bye.
Bye.
Thank you. Have a nice day. Bye-bye.